is Negin. Thank you for your attendance. Today I'm going to present my master thesis about population genetic analysis in Octopus vulgaris from Cantabrian Sea, both considering temporal and spatial genetic variations. This is my presentation outline. In the first few slides, I will introduce the work and also subject and purpose of this project, the methods we use and the analysis we have done, and following I will be showing you the results we obtain as long as the interpretation. And finally, I will be also offering some recommendation regarding the subject and also what is about to come in future. So let's start the talk by the global issue that all of us may have heard about and affected one way or another, which is unsustainable trend in fishing our marine resources. We know that marine resources are limited, and as if we go like this model here predicted, we will for sure face global food crisis and subsequent dreadful socioeconomic consequences. But the good news is that these alarming facts has risen the attention and urge that we must do something to stop this. So obviously the response to unsustainable fishery is sustainable fishing, which is vital for keeping the balance and stability of ocean and also our basic human needs like food and income. So having healthy oceans requires having healthy marine stocks, and as researchers, the first and the main step is that to assess how these stocks are doing, to have a deep understanding of marine stock status. This matter is become even more vital when it comes to species like Octopus vulgaris, which has a huge socioeconomic impact. Apparently, there is a huge demand for these amazing creatures worldwide, and especially in north part of Spain, in Asturias, as one of its major suppliers. So let's take a closer look to O vulgaris fishery in Asturias. As I was mentioned before, Octopus vulgaris has huge market value in Spain, and therefore it needs a very good and efficient and long-lasting fishery plan, because it has a huge impact on the economy of the country. At the moment, this fishery is running by a co-management system, which incorporating the politicians with community like local fishermen and all the citizens. This fishery has established like 20 years ago in form of small-scale fishery, as you can see in this map. It covers the regions from Figueras to Cudillero. All these locations are running under these co-management systems, and at the moment there are around 40 authorized boats that are fishing in these localities. There is also a seasonal closure, so the fishing activity running from December to July, and there is a 5 months gaps for stock recovery. There is also other elements like minimum landing weight of 1 kilograms and maximum catch per season. All these management systems has led to the eco-label certification, which is the first octopus fishery certified by Marine Steward Councilship in 2016, and the label renewed again in 2020. But the validation of this label is conditioned to improving the biological data from Octopus vulgaris, because at the moment there were lack of biological data when they certified this fishery. So after this relatively long introduction, here is where my master thesis contributed. So we know there are many criteria to our sustainable management that all of this has to work together. But from our part as biologists, we use genetic tools and data to obtain more biological data, from Octopus vulgaris in order to fill that gap. So as a part of a national project called Eco Seafood in Asturias, we used genetic markers in forms of microsatellites to obtain valid biological data in order to define management units, because this is critical to treat each population suitable with their needs, because if they are different populations needs different treatment, and one management plan cannot apply to all the populations. And also, we use this biological data to assessing the current stock status in these locations, because at the moment, before commencing this study, there were no biological data regarding these stocks. So we did this. We keep this approach in our mind with the final aim of improving the current fishery plans and also ensuring the validation and continuity of the label. So for this matter, we had samples from 5 locations, from here Tenerife in Canary Island, all the way to Cantabrian Sea in Asturias, north of Spain. We had also 2 temporal samples in Porto de Vega and Faro in Portugal. Overall, we had 308 individuals that all the DNAs were extracted. We have also developed 2 multiplex PCR and amplified 13 microsatellite markers distributed along the genome. Then we sequenced these samples and genotyped them using Genius Prime software, and ran different population genetic analyses based on the final data set. Okay, so now I'm going to show you the result that we obtained. These are the spatial analyses that we did. These are all for fishery season of 2020-2021. For the 5 locations that I have already shown you. So from a conservation point of view, healthy stocks refer to the stock that is genetically diverse, because it's the key element for survival in case of environmental changes. As you may see here in this graph, we assess the levels of genetic variation using observed and expected heterozygosity, considering these values are not significantly different from each other. So this graph shows us that all these populations from these 5 locations are in Hardy-Weinberg equilibrium, and also we tested for bottleneck, and there's no evidence for the recent bottleneck for these locations. Also, we analyzed allelic richness within a stock. All the 5 populations showed relatively high allelic richness, and Faro here showed the highest genetically diverse populations. So considering everything and the limited number of locations that we had, we can say that these populations are now healthy. And now move on to temporal analysis. Temporal analyses are giving us very important information, because it can tell us that either the current management plan is working efficiently, and also either these genetic results are facts or artifacts, because if they are consistent, it shows that there is actually something's happening. These 2 lines divided the regions. On the right we have Portugal, and on the left we have Asturias. Temporal samples also allowed us to measuring effective population size, And for both location, we can see the high number of effective population size, considering 50 as a threshold. Again, something interesting here is that Faro showed the ascending trend between these period of 14 years, and it can be as a result of management plans that are happening at the moment in Faro. Again, from our analysis, we can say that there is no significant threat to be considered as healthy. So the other objective was to see how many populations we are here, because if they are genetically different, they need unique management strategy. Surprisingly, in these locations from Canary Island to Cantabrian Sea, FST pairwise analyses clustered them into 4 different groups, and also this pattern tested by mental test, because we wanted to see if there is a correlation between geographic distance and genetic distance, which mental test result also was consistent with the FST pairwise results. Something interesting here is that Porto de Vega samples in Asturias here, as you can see, they are so close in Cudillero in terms of locations, but they are clustered into completely genetically different groups. Also, another interesting point here is that clustering Galicia, which is in another region in Spain, our result grouped Galicia and Asturias together. This result again showed in this slide, but when we ran the APC analysis, again same 4 groups are shown here, and we also ran a neighborhood joining tree to see to analyze them from ancestral point of view. There are 2 main genetic clusters, but then along the time they are differentiated more into more we see more heterogeneity, genetic heterogeneity. But the point here that I want to emphasize on is that clustering Galicia with Porto de Vega, as we know that there's a drastic change has been reported for this location for Galicia. So maybe that's the reason why this location is grouped along with Asturias sample, because
2 minutes.
Okay, because at the moment we don't have more samples to say if there is a fact of, or if there is because of the changes in Galicia populations. But more importantly, we have to answer if there is Asturias homogeneity, if there is genetic homogeneity in Asturias, because at the moment they are running with same management plans, and if they are different, if they are genetically different, they should be treated differently. So these results can be explained by both human activity and also environmental changes. As you can see in this graph, in 2008, there is a high fishing effort here, which can lead to drastic changes in populations afterward. Then we see changes between 2007 and 2018 populations. And also we know that sea surface temperature is increasing, and considering the short lifespan of octopus, it may also affect the observed changes. So proposing regional or local plans at the moment, it's not possible because we need more temporal data. So as a conclusion, we found that current fishery plan seems to be healthy and eco-label is valid. And we saw population cluster structuring at small and large geographic scale. And populations are in rest and genetically stable. But we need more data for proposing specific local fishery plan. I also want to mention that this study has not finished yet, and we have more samples from also Mediterranean and also from Asturias, more temporal samples, which gives us a better assessment of stock healthiness and status. And I would also like to recommend using more markers in future studies for better representment of the whole genome. Thank you for your time, and I will be happy to answer your questions.
Thank you so